Okay, so practical feedback for question 1. Let's start with A part 1. So the information tells you that there are transgenic plants which are genetically modified switchgrass. They are able to synthesize more polymers, and then the information links the ability to synthesize polymers to the growth rate of plants, which is also linked to the rate of photosynthesis. So that means, right, the transgenic plants being able to synthesize more polymers are able to have a higher rate of photosynthesis. So the question, right, asking you to compare the commercially available switchgrass versus the transgenic plant, given the same concentration of chloroplast in the extract, how will the rate of photosynthesis be the same or different, right, between these two, right, the commercially available versus the transgenic switchgrass, right? So as given in the information, the transgenic plants are able to have a higher rate of photosynthesis. So therefore, the chloroplast extract should give you a higher rate of photosynthesis for the transgenic plant. Okay, so most of you did manage to get the correct answer, but for the small number, right, who either wrote that the commercially available ones give you higher, or quite a number of you actually wrote that they will be the same, right? So this is the reason, given the information, why the thinking should be that the transgenic plants would give you a higher rate of photosynthesis given the same concentration of chloroplast. Then for the next question, right, let's look at the information and the requirement of the question first, yeah? So you are required to do a serial dilution. Serial dilution meaning, right, we are going to use SE1, which is 10% chloroplast extract, to make 1%, which is SE2. Then we use SE2 1% to make 0.1, and then using 0.1 to make 0.01. Right? So serial dilution and factor of 10. That's why it's 10%, 1%, 0.1, 0.01. And you are required to make 8 cm cube of each solution. Make 8 cm cube of each solution. Right? You don't have to consider the volume that is taken away to make the next concentration, because information is given, or in the question is written, right? Some of these will be used to make the next solution. You don't need to care about that. You just need to make up for each dilution, right? After you diluted, you should have 8 cm cube of the solution. After that, you take some away. Don't care. All right. So using the C1V1, C2V2 formula that you have learned at the beginning of last year, right, this will be the calculations that you should have done, either as a working or at least in your head, right? So I'm not going to go through the calculation. You should be okay with calculation, right? In terms of the marking points in your answer, right, it is a description question. Describe, right? Describe how you will carry out serial dilution, right? So because of that, details are needed because it's described, right? So you need to state, right, the concentrations that you are going to obtain. So instead of just saying SE2, SE3, SE4, which many of you did, right, you need to actually state what concentrations SE2, SE3, SE4 are, right? The only known concentration here is SE1, which is 10%, okay? And of course, right, in the description, right, because it's serial dilution, you need to state very clearly when you are making, say, SE2, which chloroplast extract are you going to use? So you're going to use SE1. Then when you are going to make SE3, you need to state that you are using SE2, right? Because it's serial dilution, right? So this description must be clear, okay? And because again it's a description, right, then the exact volumes needed, right, of the respective chloroplast extract as well as the distilled water needs to be stated, and the precision of the volumes needs to be correct according to the apparatus you are using, which is syringes, right? And again, as part of a description, people when reading the description should be able to follow, right? So you should state the apparatus that are required, and the apparatus should be restricted to whatever you are given, because describe how you would carry out this serial dilution, and you did that in the practical, right? Using the apparatus provided. So there's no test tube provided, for example, right? There are no small beakers provided, for example. You are provided with plastic vials, right? You have like 2 syringes, right? One is 1 cm cube, the other one is a 5 cm cube syringe. So these are the apparatus that you are restricted to in the description. Okay, so I have 2 sample answers in the answer document, right? So for those who drew the dilution table, right, for serial dilution, the dilution table should show when you are obtaining, say, SE3, for example, which concentration you are using. So you are using SE2, 1%, right? So you should have this extra column to state which concentration you are using for dilution, the second column, for a serial dilution table. Okay, then after that would be the volumes. Okay, so this is one of the sample answer. Of course, quite a number of you would it in words. So in words, this can be something you write clearly. Again, if you read the answer here, showing the concentrations of Se234, showing which chloroplast extract is going to be used for the dilution of the respective Se234, right? The volumes, the apparatus needed are all written inside here. Yeah. Okay, then move on to part 3, an advantage of carrying out serial dilution. So the advantage is that you are able to obtain very small concentrations, which is not possible with simple dilution, right? Not possible because, right, when you are going to dilute directly from the stock solution, which is 10%, if you want to obtain a very small concentration like 0.01%, the volume of the stock solution or the stock chloroplast extract will be very small, which is too small to be measured using the syringes that is available. All right, that explanation, that the part that says it's not possible with simple dilution is required in the answer. Why? Because if you don't state, right, in the comments, yeah, if you don't state that simple dilution is not possible, then serial dilution really is not advantageous, right? If I can directly dilute the stock to give me whatever concentrations I want, why not do that? Why go and do step-by-step serial dilution, right? So the advantage is really when simple dilution is not possible. If you take directly from the stock, given the very small volume of the stock needed, then you do serial, right? Because you are using a closer concentration, the previous one, right? Say for SE4, you are going to use SE3, right? Which is not 10%, right? SE3 is 0.1%. So the volume of SE3 that is needed, which is 0.8 cm cube, will be measurable, right? Using syringes, versus if you take directly from 10%, then the volume is too small, right? So this reasoning is necessary. If not, again, serial dilution really is not advantageous. It's like the same, right, as your simple dilution from stock. And I would argue simple dilution is even better because it's faster. Like, I just need to dilute straight away, right? I don't need to do step by step. Yeah. So that reasoning is very important, which I have kind of tried to at least explain in the comments. Yeah. Okay, so back to the answer, right? The second part of the answer is important. So many of you only wrote the first part, without the second part, and you do not get the mark. Yeah. Okay, then move on, right, to the importance of the cold buffer, right? The cold buffer is added when you are making the chloroplast extract. So the question also asks for importance of the cold buffer for chloroplast extraction. So note, the importance here is in terms of chloroplast extraction, not in terms of your experiment, your measurement of your results. Chloroplast extraction. Okay, so being cold allows the inactivation of whatever hydrolytic enzymes there are, because you are smashing the leaf, right? You are smashing open cells, right? So there can be, right, hydrolytic enzymes in the cytoplasm or elsewhere, we don't know, right? So if present, right, then the hydrolytic enzymes will be inactive because of the low temperature to not damage the chloroplast. Or with the pH buffer, right, then it maintains the pH to not denature the enzymes in the chloroplast. So again, note that this is for chloroplast extraction, meaning you want functional chloroplast in the chloroplast extract that you are making, right? That's why the two importance here, which point to, right, making sure that the chloroplasts are intact, the enzymes in the chloroplast are working, so that your chloroplast extraction is useful. All right. Okay, so it's quite a number of you, right, for the cold, the low temperature, right, mentioned that the low temperature is to stop the enzymes in the chloroplast from working, right, from having photosynthesis. So first of all, that's not related to chloroplast extraction, right? That's more related to the experiment. And if you think about the experiment, so what if the chloroplast extracts are having photosynthesis, right? It doesn't affect the results because you have not put in the agar cube yet, right? Make sense? Yeah, so even if at this stage, when you are making the extract, when you are waiting, cutting the agar, if there is photosynthesis, it doesn't really matter. It doesn't affect the results. Yeah, so the reason for the cold is really not to inactivate the enzymes for photosynthesis, right? It's more the hydrolytic enzymes to prevent damage to chloroplasts. Okay, then for the pH, right, I did not put it inside the comments. For the pH, again, a lot of you mentioned that the pH is to be kept optimum for the action of the enzymes for photosynthesis. So again, that is not related to chloroplast extraction, right? That's more for the reaction, more for the experiment later, right? So again, therefore the pH here is simply to prevent denaturation. So the chloroplasts have functional enzymes, so the chloroplast extract is useful, right, for chloroplast extraction. Yeah. So when you are in the A levels, when you are coming up looking at the questions, can you read the questions carefully and look out for what the boundary, what is the context of the question? Right, so here, chloroplast extraction is the boundary, right? Your importance needs to be related to chloroplast extraction, not experiment, right? Not the reaction. Yeah. Okay, then next, for the drawing, right, most of you got this correct, obviously. For the few who lost mark, right, please follow instruction and don't be lazy. The instruction says to add root line, ruler, right? So use a ruler, and the labels are actually spelled out for you. So why take shortcuts, why leave out certain words, why use short forms, right? It's spelled out in the instructions. Follow instructions. Okay, move on. to the table. This one is quite disappointing because a lot of you forgot the basic requirement of drawing a table. The independent variable should be on the leftmost column. So again, independent variable on the leftmost column. The independent variable in this experiment is the concentration of the chloroplast extract, not tube number. Right, so SC1, SC2, SC3, SC4, they are not concentrations, right? They are like names of the extract which have different concentrations. State the concentrations as the independent variable. Secondly, right, also, quite a number of you forgot that should be recorded to whole number. We have done this in the term 3 revision, in some of your practicals. Okay, so I'm not going to explain why it should be whole number. Okay, just remember that recorded by or measured by a ruler should be in whole number. Okay, a few of you didn't even read the instructions and recorded to cm. That is extremely disappointing. Right, it's clearly stated, right, to record to the nearest mm. Okay, then a few of you got marked down by the trend. Okay, so the trend should be, right, with increasing concentration of chloroplast extract, the length of the stained region should be smaller, right? So again, increasing, right? Increasing concentration, higher concentration of chloroplast extract, more photosynthesis, right? So more decolorization, right? So the length of the stained region should be shorter. Okay, and for TE, the unknown, the correct results you should get should be either same as 1% or same as 0.1% or in between 1% and 0.1%. Right? Okay. So now using the table, right, using the results, estimate your unknown, right, TE. Okay. So I'm just gonna repeat something that should have been like mentioned in lessons, in case you forgot. When you are doing such estimation based on standards, right? So the SC1, 2, 3, 4, the 10%, 1%, 0.1%, 0.01%, these are known concentrations, right? You use these 4 results of known concentrations to compare, right, with your TE. So the 4 known concentrations are like standards, right, for you to estimate your unknown. Whenever you are doing this, right, whether it is in this case, you know, your Agar cube, whether it's what you are more familiar with, right, with some glucose standard, Benedict's test, whatever it is, right, whenever you are comparing to standard, your estimation either state the closest standard that is or state the standard that is closest to your unknown, or if it is right smack in the middle of 2 standards, then just give your estimation as a range between the 2 known concentrations, the 2 standards. Do not go and try to calculate, say for example, between 0.1 and 1%, right? Then some of you put 0.5, 0.4. All these are not science. They are not evidence-based. They are your estimation. They are your prediction, assuming that the change in length is proportional to the change in concentration. All these are assumptions, right? Not evidence-based, not science. So estimation, either estimate to the closest standard, or if it is really in between, then just give your estimation as in between. Okay, so the answer, if you have gotten the correct trend for this question, should be either 0.1% or 1%, or between 0.1% and 1%. Really, these three answers if the trend was correct, if your TE's result was correct. All right. So please remember this, because this is not the first time I have seen you make this mistake. Okay, so please remember this. Okay, then next for the just one significant source of error. So if the question asks you for one significant, then please give the most significant source of error, right? Okay, after that, describe the improvements. Describe, right? So remember, describe requires details. Yeah. Okay, so the one most significant error, source of error, should be the light, right? The source of light, because there was no lamp given, right? And we are using the ambient light, whatever light that's available in the lab, which is not constant. So, and because this is photosynthesis experiment, right? That obviously light is a major factor affecting the rate of photosynthesis and therefore affecting the results, right? So since this is just a major factor which is not controlled, then obviously it is significant, right? So really, that should be the first thing you think of, right? Photosynthesis, light, okay? I also accept that temperature being another very important factor affecting photosynthesis, that one of the tube, the TE unknown, is not of the same temperature as the rest. The rest are all room temperature, but TE was made with cold buffer, right? So it is not of the same temperature as the rest. Okay, so that would be acceptable as an error. However, right, if you look at the comments, if what you are saying is all the tubes are not controlled in terms of temperature, there's no water bath for all the tubes, then that is not accepted, because if you have read the information in front, right, there is this instruction saying that it is not necessary to control the temperature. The room temperature of the lab is assumed to be constant. Right, so for SE1 to 4, which are done under room temperature, it is assumed to be constant already. So if your source of error is that SE1 to 4 temperature is not controlled, then that is not acceptable. All right, that's why if you look back at the answer only mentioned TE being colder, because TE is not at room temperature because of the cold buffer. So that is not therefore constant, or not the same as the rest. Okay. Other not accepted suggestions would be that you are talking about your own mistakes or your own shortcomings. Okay, so quite a number of you mentioned difficulty in cutting the agar to the correct size, right? So that is saying that you cannot follow instructions. That is not a source of error of the experiment. That is your fault. Or some of you mentioned, right, you did not wash the syringes when you are transferring your chloroplast extract between different vials. Well, that is really your fault. So things like that shouldn't be considered, or you shouldn't suggest your own shortcomings as source of error, right? Critique the experiment, critique the procedures, not yourself. Okay, and again, I mentioned, right, because the question asks for one significant source of error, please give the one most important, most significant source of error. So color in this case is not the most significant, because we are not dealing with different colors. We are not assigning different colors, different shades of blue, darker blue, lighter blue. It's just presence or absence of blue. That is really not subjective. Okay. Then others lah, so I leave you to read some of the others. Okay, move on to this quite important comment here. For those who blindly mention that color is a source of error, and also blindly suggest colorimeter as the improvement. I say blindly because if you think about it, it is not feasible to use a colorimeter even if color really is a significant source of error. Why? Because we are not dealing with liquid samples, right? We are dealing with this agar cube that cannot be put into the colorimeter. Also, we are not measuring color intensity here, right? We are measuring the length of stained region. How to use a colorimeter to measure the length of the stained region? You're going to scoop up different parts of the agar cube, put the different parts into the colorimeter, and see which part of the agar cube gives you a colorless reading. No, right? Can't do that, right? So to measure the length of stained region in a solid sample, colorimeter is simply not feasible if you think about it. That's why I say for those who suggested colorimeter, you did it blindly because you saw color and therefore blindly saying that it is a source of error, then blindly suggesting colorimeter. All right. While that shows you maybe have revised and studied some of the previous practical, you are not thinking, right, in this context, in this practical. So that is not something that you should be doing or not doing in terms of thinking, right, in the A level. Okay. So again, the question says describe, right, improvement. So details, details. Okay. So distance from the lamp, right? If you mention temperature, what's the temperature? Duration for equilibration of temperature, things. These kind of practical details are needed for such improvements. Okay, then I move on to my last part, the maths question. And honestly, a primary school kid can do this, but you can't, so that is a bit alarming. Right, so you are asked to calculate the percentage decolorization, right? Percentage decolorization. Your agar cube is measured at 1.5 cm or 15 mm long, wide and high because it's a cube. At first it is stained throughout at first, right? Now it is mentioned after the experiment, instead of 15 cm or 15 mm length stained, now the length of the stained region is 10 mm, right? So from whole cube stained, now the center of the cube, which measures 10 mm, not 10 cm, typo, 10 mm, right? Length in the center of the cube is stained, right? The rest is colorless, not yellow. No, the picture looks yellow, but it's colorless, right? So how do you then find the percentage decolorization, right? You find the volume of decolorized region, right? Volume, right? It's a cube. Right? So how do you find the volume of decolorization? You find the total volume, you find the volume stained, then you minus, right? Then with that, with the volume of decolorization, right, you take that volume of decolorization over the total volume, find the percentage, that will be your percentage decolorization. Not some team maths, right? It's a cube. So for those who use only a length, a few of you use area, I don't know for what reason, right? It's a cube, use volume, yeah? Okay, so I'm not going to explain the calculation. The only thing I'm going to say for the calculation is calculation should be step by step. Don't skip step. Don't put things together in one big formula. Do things step by step. Label what each step is, like you see here in the answer, right? So when someone reads it, when you read it, you know, right? The train of thought, you know what the calculations are, you know what each number stands for. That is what is called clear working. All right? Don't be lazy, don't skip. Don't just have one formula and then suddenly the final answer. Yeah? Then for the final answer, right? Because your raw data is your length, right? Whether it is 15 mm for the total volume, 10 mm for you to calculate your stained volume, right, is in whole number. So your final answer should either follow the precision of the raw data, which is whole number, or because it's calculation, you can add one decimal place. So it's either whole number or one decimal place. State which one you rounded off to. Must state, because if you don't state, then it seems like you have miscalculated. So you must state that you rounded off to whatever precision, right? And for those who keep forgetting or mix up with chemistry, right, we don't use SF. This is from the Cambridge examiners. Precision should be looking at decimal places. Unless otherwise stated, use decimal places for bioprac. Okay, I don't care what you do in chem, yeah? So for the many of you who simply put their rounded off to 3SF, even if your final answer is the same, right, because of the 3SF, you may be marked down by the Cambridge examiners. Okay, so take note of that. Okay, so that's my part, all right?